Good evening, everyone, and welcome to today's class. We'll start in 5 minutes as usual so that people can join in. Okay? can start our session today. So, little bit of recap from the last class what we did. So, we are into membrane protein biogenesis. Before that, we actually saw how membrane proteins perform their function of maintaining the electrochemical gradient across the membrane. So one of the example which we have seen was the sodium potassium ATPase, and we also understood how, or we learned about that why maintaining a particular type of electrochemical gradient is necessary for the survival of the cell. And the major function of transportation, it is accomplished by simultaneously maintaining the gradient, and these gradients are easily maintained by transporters, and transporters, as we have learned, are of different type. For example, if we talk about transportation, it can be passive and active, right? So all this, though for our own understanding we classify them as active transport or passive transport, but in reality they are working simultaneously to assure that the electrochemical or ionic concentration across the membrane is well maintained. Okay, so this is one of the reason we also study about the concept of buffer, right? If you remember in your early chemistry classes, you guys might have learned about buffering or buffers. Our blood is a very good buffer system, right? The moment we eat something, the moment any solute concentration changes, our whole system is so adept and so well synchronized in maintaining the homeostasis of our internal system, right? So for that you need active, or here I don't mean active means where the energy is associated, I mean active, the functional form of transporters or membrane proteins or anything which is communicating, which is able to communicate the external environment to the internal environment of a cell, that should be very precise and very active, correct? So for that purpose only we studied about the transportation. Then we switched some of our gears, and then we understood, though we now understand that these transporters are important at the end of the day, right? And these transporters are proteins, correct? We understand basic translation, right? Replication, transcription, translation. We understand about the cytosolic proteins also, but we don't know how these proteins are assembled into the membrane, how they are taken place from the ER to the membrane or to other organelles, right? Because every organelle, we are eukaryotic cells, right? Eukaryotic cells are compartmentalized cells, correct? And each compartment having a membrane might have proteins associated with them. There are proteins which are needed in the cytosol, as well as there are proteins which are associated with the membrane. So how come the system knows where the protein should go? Either it should go to the plasma membrane, or it should go to the mitochondrial membrane, or it should go to the lysosomes, or it should go back to the ER or to the nucleus or to the nuclear membrane, all those things, right? So we have seen some of the glimpses of secretory pathway in the eukaryotic cell excuse me, correct? And so continuing on this series of secretory pathways and how these proteins are actually getting assembled into the membrane, not all those proteins which are present in the membrane. That membrane can be from any origin, right? It can be a plasma membrane, it can be a mitochondrial membrane, it can be a lysosomal membrane, it can be a ER membrane, it can be a nuclear membrane, any membrane where protein is associated, correct? So very seminal work has been done in this particular field, and almost 4 Nobel Prizes have been awarded for the discovery of secretory pathways and how proteins are translated, especially the membrane proteins, right? So you can guess how important and basic this functionality is for the living cell, okay? So Camillo Golgi, as you might have understood from the name itself, that Camillo Golgi, Santiago Cajal, in 1906 they got the Nobel Prize for recognition of their work on the structure of the nervous system. So what their work was, this structure of the nervous system, they actually associated the transmission of proteins which are accumulated in the synapses, right? So there are, if you remember your basic neuron structures, in the axon at the end there are axonal bulbs, right, where you have signaling molecules stored in the vesicles, correct? So they actually showed how these things are stored at the synapses. Then Albert Claude, Christian Dubé, they and Palade, they got Nobel Prize in 1974 for their discovery and function of the organization of the cell. We might focus a little bit more on Palade's work because his work was very, very, very pioneer in the secretory protein synthesis and how they are translocated and transported within the cell. Okay. Then Blobel got his Nobel Prize in 1999 for the discovery that proteins have intrinsic signals that govern their transport and localization on the cell. Okay. Then we have Sheckman and Rothman, and they got their Nobel Prize in 2013 for their discovery of machinery regulating vesicle traffic, a major transport system in our cell. So you can see that Palade's student was Blobel, right? So Blobel actually continued the work of Palade in this field. So we're gonna see mostly how both of them actually pioneered in the field and got us to the understanding how secretory proteins are actually assembled in the membrane, or how they are transported within the membrane. So the best example for you guys to understand, for example, you have soap bubbles, right? We all have seen soap bubbles. So if we are talking about the transportation of proteins within the cell, so you need to be very clear about the basic biochemical pathways. So let me remind a little bit of it so that you can understand the compartments which are majorly associated. So for example, this is your basic cell, right? This is your nucleus, this is ER, and finally these are your Golgi bodies, right? And this is your cytosol, correct? And this is the membrane. This is the membrane where proteins are there. So replication, transcription will take place here, right? And then ribosome will assemble on the ER for the protein synthesis, correct? For the protein synthesis. So this protein, whether it needs to stay in the cytosol or it needs to go to the membrane, it will be decided right here. This is what we know right now. But how they are transported from nucleus to the RER to the Golgi, and from Golgi to the other places, right, wherever it needs to go. So all this happens via vesicles. So what are vesicles? Vesicles are the small lipid membranous structure which are pinched off, right? Because if you want to transport anything, for example, something being synthesized here and it needs to be taken here, so what will happen? A membrane will be pinched off from here and it will merge with the Golgi apparatus, and hence the cargo will be delivered. Better representation, I am not good at drawing, which you can clearly see. A better representation you can see here, right? So this is rough ER where protein is being synthesized. Now it needs to go to the Golgi for sorting purposes. So how it will go? It will go via vesicles. So these vesicles are equivalent to the bubbles which we are representing here. Okay. So this is a very brief overview of the intracellular transport. So a thing which I want you guys to understand is soap bubbles, if you are not aware of it, they are single membrane structures, right? But if we talk about the vesicles which are in biological system, they will be double membrane, because we have seen, right, plasma membrane is lipid bilayer in nature, correct, indicating that even the vesicles will be bilayer in nature. So how this happens? And from a previous lipid discussion, we also know that there are characteristic differences between the inner leaflet and the outer leaflet. That is, the lipid bilayer which is facing the cytosol and the lipid layer which is facing towards the extracellular matrix, they are different, meaning they are asymmetric in nature, right? So if you can see here, the orange one is the membrane which is facing towards the cytosol, and the black one or the gray one is the one leaflet or membrane which is facing towards the extracellular matrix. So when the vesicle is formed, right, so vesicle will be pinched off from the organelle. For example, if you are talking about ER, so when it is being pinched off, the outside of the membrane will be same as the membrane which is facing the cytosol, right, and the inside of a membrane will be same which is facing the extracellular matrix or the lumen, correct? So which is pretty evident from this diagram. Now this is a very simplistic way of understanding how intracellular transport takes place. So as you can see, the red arrow indicates gated transport. So what will be the gated transport? Gated transport means it is mediated with the help of proteins, correct? Transmembrane transport means the transportation is taking place with the help of transmembrane transporters or integral membrane transporters. The green one is the vesicular transport, meaning the bilayer or will be pinched off from the membrane, and that membrane will have to fuse to its destination for releasing of the cargo or the substrate molecule, whatever we are transporting at the end of the day, right? So you can see from the cytosol itself, if from the cytosol something needs to go to the nucleus, it will be highly regulated process, right, because not everything can enter into the nucleus. So it is primarily mediated by the gated transportation, while something from the cytosol, or especially proteins we are talking about here, right, if it needs to get transported to other organelles such as mitochondria, plastids, peroxisomes, endoplasmic reticulum, it will be via transmembrane transporters, okay. Then from endoplasmic reticulum to the Golgi body, then further to the secretory pathway. This you can easily see that this is the part of the secretory pathway, correct? So all the secretory pathway is dominated by the vesicular transport. Hence, understanding the vesicles itself and how proteins are being protected from the outside when they are being transported towards their destination is very important. So a basic thing which we can ask, for example, there will be a lot of proteases in the cytosol, right? Because proteases don't know where to go. Wherever they find a nascent protein, they'll try to target it, correct? So how they are protected? Because a fresh protein is being synthesized here. Now for the sorting purposes, it needs to go to the Golgi. So when it is traveling from ER to Golgi, the nascent protein will be subjected to proteases action, correct? But it doesn't happen, because if it is happening, then no protein will be secreted at all, and no protein will be associated or get embedded in the membrane, right? It is not happening, we know this. So why it is not happening? Because there is existence of intracellular transport, primarily via vesicular transportation, okay. I hope that I am able to emphasize more on the point that most of the intracellular transportation is a type of vesicular transport at the end of the day, okay. So I want to bring your attention to the very basics of membrane. Now why I want to reiterate myself here? Because as I have just mentioned, that majority of the intracellular transportation is via vesicular transportation, right? Vesicles at the end of the day, what are they? They are the membranous structure. And since the beginning of this course, we are understanding the importance of membrane, the components of membrane, their functionality, their structure, their physical properties, their chemical properties, right? So if we are talking about major transportation within the cell, they are of vesicular type, and hence vesicles are a type of membrane. So, and we know the membrane model system, the most accepted model will be the fluid mosaic model. As we have discussed earlier, that there were plenty of experiments which indicated that the membrane is of fluidic in nature. Now you should understand from the perspective of transportation why membranes need to be fluidic in nature. Because, for example, if we talk about these bubbles, if these are very stringent bubbles, they won't fuse, right? But on the other hand, the major type of transportation, vesicular transport, meaning the membranes have to fuse, as you can see from here. It is pinching off, correct? It is also fusing in here also, right? So if the membranes are rigid in nature and they are not flexible, or in membrane biology terms, if they are not fluidic in nature, they will not function well for the intracellular transportation or the vesicular transportation, which we are calling it. So how we know that the membranes are fluidic in nature? One experiment was where when two different membranous system, one from the mouse cell being decorated with one type of fluorophore, merged with the human cell, this membrane was decorated with other type of fluorophore. When they were combined together, after some time, after incubation period, we could see that the heterokaryon which was formed, it was having both the type of fluorophore, indicating that though we have merged two different cells which are having two different fluorophore, after some time there will be mixing of these fluorophore, indicating that the membrane is actually dynamic in nature and fluidic in nature. Okay. Second experiment which actually supported this phenomenon was FRAP. FRAP is basically fluorescence recovery after photo bleaching. So if we follow the nomenclature of this assay, fluorescence recovery after photo bleaching, right? So we can get the idea about the whole experiment. So what FRAP does is, so there is a membrane, right, and we have labeled it with some fluorophore, correct, or fluorescent molecule. When you point the laser on this fluorophores to a certain area of the cell or the membrane, what will happen? They will lose their fluorescence. Hence, when you observe them, there will be no light from this particular section. There will be a darkness, correct? But after some time, when you notice the same place, what will happen? That they will regain their fluorescence. As you can see, once the light was shined on that particular area where we want bleaching effect, then there will be loss in the fluorescence. But after some time, what will happen? The fluorescence will recover gradually. So what it indicates, that if we have bleached this particular area, after some time this area has been recovered from the surrounding fluorophores. That is the only possible reason, right? That it will be covered by the nearby fluorescent labeled proteins, and hence we can again detect the signal, correct? So this also indicates that our membrane is in fluidic in nature, correct? Another way of being sure that our membranes are fluidic in nature is via the opposite of FRAP. So what will be the opposite of FRAP? The opposite will be fluorescence loss in photo bleaching, right? So what will happen is fluorescence loss. It means from where we have created the bleached area, right? So after some time, that bleached area will be recovered from the surrounding fluorophore molecules. Hence, if it is being surrounded from it has been recovered from the surrounding fluorophore molecule, then the signal from the surrounding fluorophore molecule should decrease, Correct? So this is what the scientists have observed, indicating and proving more and more that these membranes are fluidic in nature. Okay, so if we talk about, we are saying that a lot of transportation inside the cell is a vesicular type of transportation, right? So imagine this, that we are blocking this secretion. So according to your, even your wildest imagination, what will happen, right? Think about it. When proteins are being synthesized on the ER, they are being sent to the Golgi, and then the Golgi decides, depending upon certain mechanisms in place, where that protein needs to be circulated or targeted. Correct? So imagine if this process is not in place, then what will happen, and how we can discover these processes. Correct? I guess, okay, yeah. So we'll see one small representation of the same thing which I'm talking about, and then I'm gonna discuss this figure with you guys. So just give me 2 minutes so that I can load this.
Here,
Yeah. So this video gonna give you a brief about how the vesicular transportation, especially the proteins which are supposed to be inserted in the membrane, are secreted or synthesized. I hope that you guys can hear this.
here we'll learn how transmembrane proteins import into the ER and embed themselves in the ER bilayer. To begin, start a table to learn the 2 types of proteins that enter the ER. Denote that they are water-soluble proteins and transmembrane proteins, which add to the ER's phospholipid bilayer. Both types have signal sequences that direct them to the ER. Here we'll focus only on transmembrane proteins, which import co-translationally. Ribosomes continue synthesizing them as they cross the membrane. To begin, return to our table to learn the different types of membrane proteins. Denote that they include the following: type 1 membrane proteins, which have a signal sequence at their N-terminus and an internal stop transfer sequence. Type 2 membrane proteins, which have an internal signal sequence instead of a terminal one. Type 3, which also have an internal signal sequence instead of a terminal one. We'll distinguish them from type 2 proteins shortly. Type 4, which are multipass membrane proteins that have multiple hydrophobic regions embedded in the bilayer. We'll focus on a 2-pass protein, which has an internal signal sequence and a stop transfer sequence. We'll illustrate how each of these proteins inserts into the membrane. Let's start with type 1 membrane proteins. First, draw an ER membrane with a translocon and a membrane-bound signal peptidase. The translocon is a channel through which proteins enter the ER lumen. Label the cytosolic and luminal sides of the membrane. Next, draw a ribosome bound to the cytosolic side of the translocon. Show that it's translating an mRNA transcript. Now draw a nascent protein emerging from the ribosome and entering the translocon. This is known as co-translational import. The protein threads through the translocon as it's synthesized. Show that the protein has an alpha helix domain and a signal sequence that binds the translocon. The signal sequence functions as an address label and directs the growing protein to the ER membrane. Label the alpha helix stop transfer sequence. It's an internal sequence that is hydrophobic, just like the signal sequence. Indicate that this is step 1. The stop transfer sequence stops the transfer of the nascent peptide through the translocon. Note that stop transfer sequences are always hydrophobic transmembrane domains. In step 2, indicate that signal peptidase cleaves the signal sequence. Step 3, show that this allows our stop transfer sequence to exit the translocon laterally. Even after this occurs, the ribosome continues translating until the remainder of the protein is synthesized before dissociating from the membrane. Finally, label the luminal side of the protein as NH2, which is its N-terminus, and the cytosolic end as COOH, which is its C-terminus. Now let's illustrate type 2 protein insertion. This time draw a cytosolic ribosome translating an mRNA transcript. Show that the emerging protein has an internal signal sequence which functions as an address label, just like the terminal signal sequence in our type 1 diagram. Label the protein's N-terminus. Now show the ribosome, mRNA, translocon complex in the ER membrane. Illustrate that the internal signal sequence acts as an alpha helix domain and binds to the translocon. This transmembrane alpha helix anchors the protein in the membrane. Finally, use an arrow to show that, much like the stop transfer sequence in our type 1 diagram, the internal signal sequence exits the translocon laterally in the membrane. Indicate that this time, signal peptidase does not cleave the protein. Finally, label the C-terminal and N-terminal ends. This membrane topology makes this a type 2 protein. Now let's show how type 2 proteins differ from type 3 proteins. Draw another portion of the ER membrane with a translocon embedded within it. This time, draw the protein and alpha helix domain in the translocon first. Note that so far this diagram is identical to our type 2 diagram. Here, draw the ribosome mRNA transcript on the right side of the translocon. Now label the emerging end of the protein as its N-terminus. Show that the alpha helix domain exits the translocon laterally. Again, no cleavage occurs here. Now label the N-terminus and the C-terminus of the protein. It has the opposite topology of a type 2 protein. Let's use a stitch analogy to clarify this concept and introduce type 4 proteins. Draw a needle tied to a spool of thread in the cytosol. Label the needle internal signal sequence and the needle point N-terminus. Label the spool ribosome. Begin with type 2 insertion. Draw a line to represent a piece of cloth. Imagine the cytosol above it and the ER lumen below it. Now imagine that we put one stitch into our cloth. Show that our needle, the internal signal sequence, remains inserted in the membrane and that the needle point, the N-terminus, emerges into the cytosolic side. Now imagine that we pull the remaining thread from our spool through the cloth. Indicate that the free end of the thread in the ER lumen is the C-terminus. Next, let's use the same analogy for type 3. Imagine that we use our needle to stick the cloth just once, instead of making a full stitch. Note that the needle points in the opposite direction of our type 2 needle. Illustrate that we unwind the thread from our spool. Now show that the C-terminus remains in the cytosol and the N-terminus is in the ER lumen. This brings us to type 4 proteins. Let's use our thread analogy to illustrate one now. First, show that just like our type 2 protein, we thread our needle through the membrane to form a stitch. This time, label the portion of the thread that crosses the membrane stop transfer sequence. Just as in type 1 protein insertion, stop transfer sequences stop the transfer of protein through the translocon. Show that this time we do not pull the rest of the thread in our spool through the cloth. Label the terminal ends of our thread such that both the C-terminus and N-terminus remain in the cytosol. With that imagery, now we can draw a final ER membrane to illustrate an actual type 4 protein. Draw two alpha helices that traverse the membrane. The left helix is a stop transfer sequence, and the right is an internal signal sequence. Draw a line to connect them in the ER lumen. Show that both the N terminus and the C terminus extend into the cytosol. Indicate that this is a two-pass transmembrane protein. Type 4 proteins can pass through the membrane many more times. They're not limited to 2 passes. Write that for more than 2 passes, each stop transfer sequence must alternate with a start transfer sequence that restarts the transfer of the protein through the membrane. We'll not draw this here. This concludes our diagram.
So it might be complicated at first, but I will highly recommend if you guys can just go back to this particular video again. So I'm gonna come back to my slides and we'll see this in somewhat simplified way. Okay, so whatever you have learned,
Ma'am, can you please share the videos link?
the thing is I will be uploading it on the Moodle platform. The thing is I'm having some issues for uploading the videos. I might do this by the end of this week. I'll upload all these videos and the slides also, so you can just copy paste these links from the slide itself. Is it okay?
Okay.
Yeah.
Yes, please.
Maybe I can do the slide upload. I'm having trouble in uploading the videos, but I maybe the lipid part and the protein part also I'll upload the slides as soon as possible. And
Sure,
I'm not
thank you.
able to admit this person, Shubham Kalmakar. I'm trying, but it is not accepting. I'm sorry, Shubham. Yeah. So so whatever we have seen, it might be too much information in short time, right? But if you just take the zeal out of those processes, so what do we want? We want intracellular transportation.
And there is the option of admitting coming from my side. Should I admit him?
Yeah, I'm also trying to admit him. You can try if it is possible. Because I'm trying from
No,
the-
ma'am. No.
yeah, there's some error, I guess. It's okay.
Yeah.
I maybe drop a text. I don't think so he can see our chats also. It's okay. There might be some error. Yeah. So the overall explanation, what I want you guys to understand from that very brief video, you don't need to worry about all the proteins associated with it or the names and the terminologies. What I want you guys to understand and imagine is that, imagine it's a cell needs to communicate from the outside to the inside, as well as within the cell also there needs to be some communication, right? Because then how would we know whenever the stimulus comes the cell needs to respond? This response is in this way only. There might be increase in some protein synthesis, there might be some decrease in protein synthesis, there might be increase in concentration of certain ions, and vice versa. So all this happens when a stimulus comes. So when the stimulus is coming from the outside, and most of the time it will be from the outside itself, right? So when the stimulus is coming from the outside, the cell needs to prepare itself to react from that action. The action was the stimulus. So how it will react? It will react by, suppose if the signal was to differentiate the cell or for multiplication, which means we need to increase the production of certain proteins, or we need to decrease the production of certain proteins which are not state specific. So in that time, during that time, there has to be a very synchronized communication within the cell also, correct? And that communication, if you just compartmentalize the cell, so this is your basic secretory pathway. So what will happen is your ER will be there, and your basic cell biology dictates that the protein is synthesized on the RER, correct? So when you are synthesizing the protein on the RER, then the protein needs to go to the places where it is needed. It can be in the cytosol, it can be in the mitochondria, it can be in the chloroplast, it can be in the nucleus, it can be on the membrane. But the protein does not have any memory that where it needs to go. This is dictated by signal peptides. So what are signal peptides? It is just like, for example, if you talk about the post office. So when you go to the post office, you put the zip code or pin code, right? You put the pin code in it. So pin code basically tells the letter or the postmaster that where this letter needs to go. Similarly, if we have a vesicle which has protein in it having a particular signal, the proteins which are there in the surrounding, which will take this particular cargo to the destination, they know where the destination is, correct? So the signal peptide, the function of signal peptide is just to tell the cell that where this protein needs to go. All those type 1, type 2, type 3, type 4, these are just the permutation and combination of this basic phenomena only. Okay, so I hope that it is bit like you can imagine what I am trying to say. There are multiple proteins synthesized at all the time, correct? Oh, this request is still there. Okay, correct. So how does a cell know whether this protein is needed on the membrane, on the mitochondria, or which membrane at the end of the day, or whether this protein needs to be secreted outside? So all this is governed by proteins which are there in the cytosol. But to the proteins, how do the proteins know where that particular cargo needs to be targeted? This is done by signal peptides or targeting sequences. Okay, it is all about complementation. So if you have a target, if you have a pin code, the postmaster will know where to send this letter. Similarly, if you have a target sequence, if you have a signal peptide, the proteins in the cytosol or in the cell will know where we have to send this particular cargo or protein. Okay. So so this is the overall view of the secretory proteins. I am re-emphasizing it multiple times so that you guys can have basic imagination and understanding of how the transportation of proteins occur inside the cell, because most of the time the notion which we have, at least I had before understanding the secretory pathway, is that proteins will be synthesized in the cytosol and they will stay there. But I never thought that how they are being assembled into the membrane. So it's actually a very, very delicate machinery, and a lot of proteins and cofactors are involved in making sure that the cell has all the required protein in the right amount and at the right place in the right time. Okay, so again, just the simplistic view of secretory proteins is that we'll have rough ER, where there will be synthesis of the proteins in the lumen. Then after this, there will be ejection of these vesicles filled with these proteins, and when they are translating, if they are secretory in nature, they'll be pinched off from the membrane, and depending upon the signal peptide, they will be either digested if the protein is not folded properly or there are certain issues with it, or some things will be added if the protein is not proper, then ubiquitin will come to it and all those secondary pathways will take place. So this is the way how we actually knew that proteins are secreted in a vesicle. So what will happen is, if you have a vesicle and a protein is being translated on it, so if you add a proteases, so proteases will be from the outside, so it won't have any access to the protein which is being synthesized, hence it won't be able to digest it. On the other hand, if you have a vesicle and you add the protease, if you don't have the vesicle and you add the proteases, right, what will happen is the protein will get digested because of the proteases. So to show this in experimentation or in vitro, what happens is, when we take the vesicles, or these are called microsomes, when we take the vesicles which are having protein translated in them, if we add detergent, so what will happen if you are adding detergent? The detergent will disrupt the membrane, because membrane at the end of the day is what? Is lipidic in nature, correct, if you remember, and it is a culmination of non-ionic interactions. Detergent is somewhat similar to these lipids, and it disrupts these weak ionic interactions, and hence the protein will be there in the solution, and when you add proteases, it will degrade the protein. On the other hand, if you don't add the detergent, the vesicle will be intact, the protein will be intact, and even if you add proteases, the proteases won't be having any access to the protein, and hence it will stay functional, or there will be no digestion of the protein. Correct. This is how we have shown via experiments that whatever Palade did. So as I've mentioned, the two scientists name, Palade and Blobel, they are the pioneers in understanding the secretory pathway and designing sophisticated experiments to understand how the secretory pathway works and how we can actually for sure say that proteins are actually transported within the cell via vesicles. In the coming years, we have also seen that even there are RNAs which are being sent outside the cell in the form of vesicles only. Though the major mode of transportation within the cell is vesicular transport, but there are RNAs also which are being transported outside the cell via these vesicle system only. Okay, so I guess this is it for today. We might go ahead with this, but I feel like there's a lot of information packed here, so you guys should take a little bit of time and read about it. If you have any doubt, please feel free to drop me a mail, and we'll continue this, like we'll continue our discussion in the next class. So thank you so much.